Hello everyone, this is our lecture over Chapter 17, and this is a connection really with Chapter 16, which was all about DNA and how DNA is really the genetic material in the instruction booklet, whereas Chapter 17 is really where all the real work happens. So that DNA is going to be converted into RNA language, and that RNA language is going to be used to build the proteins that make up our traits. And so there's one more group of scientists that I want you to practice, and that's Beadle and Tatum. And what Beadle and Tatum's hypothesis was is that each gene codes for one enzyme. And so they specifically worked with mutants of mold. And so in this case here, the wild type, remember wild type from the fruit flies, is normal, what you'd normally find in nature. And so the mutant forms of the mold are ones that have some kind of a defect in the gene that codes for the enzyme. And so as you can see here, the class 1 mutant enzyme A is knocked out. And so in that case, this precursor starts to accumulate because it doesn't have the necessary enzyme to convert the precursor to ornithine. And so this starts to increase in concentration and the pathway shuts down. The class 2 mutants just have a mutation then in a different gene, and that particular gene they found out codes for this enzyme, and so in that case ornithine would start to accumulate in excess because the pathway can't go any farther without enzyme B, and so on. Really, they were on the right track, except we know that DNA codes for proteins, and proteins have more than just enzyme functions. And so really the better hypothesis is one gene, one polypeptide. And we'll even see some exceptions to that in the next chapter as well. So to get started, to compare the bacterial cell, the prokaryote, to the eukaryote, you'll see the basic central dogma is happening. And so remember, the central dogma from earlier in the semester is DNA to RNA to protein. And that pathway is going to proceed really in the cytoplasm for the prokaryotic cell, and specifically the DNA to RNA step is called transcription. That's transcription. And you'll notice then that transcription happens in the cytoplasm. That's the DNA to RNA, specifically messenger RNA is what we'll be talking about. And then that RNA is going to go and find a ribosome and be used to sequence the amino acids together to build the protein. And so that is called translation. That is translation. And these same steps are going to really happen in our cells as well, being being a eukaryote. The difference is that transcription is going to happen, of course, where the DNA is found, which is in the nucleus. And then translation happens out in the cytoplasm, where the ribosomes are found. Again, those ribosomes could be just found freely floating. They could be also bound to that rough ER or even bound to the nuclear membrane. You'll notice an extra little step in here. Okay, that is another thing that sets eukaryotes aside from the prokaryotes. And so there's an RNA processing step that happens only in eukaryotes. And again, that is going to happen still in the nucleus of the eukaryote prior to it leaving the nucleus and being used to synthesize that protein. Okay, so make sure you practice the difference between the prokaryote here in the bacteria and the eukaryote. Okay, so first of all, the genetic code. We've known about the genetic code really since the 1960s. And so the instructions are really written in these 3-letter codes. Okay, so it's going to take 3 nucleotides to code for 1 amino acid. Okay, so again, the 3 is 3 nucleotides for each single amino acid. Okay, And so you can kind of see that here in this diagram. Again, here's the central dogma. Okay, here's the DNA to messenger RNA to protein. Again, DNA to RNA is called transcription, and RNA to protein is called translation. And you'll see if I kind of draw a line down, for each group of 3 in the DNA, it's going to code for a group of 3 in the messenger RNA. We call that a codon. Okay, each group of 3 in the messenger RNA is a codon. And then that single codon is going to code for that one amino acid. So again, here's our 3 nucleotides, or the triplet that makes up the codon. And then that gives us one amino acid. Okay, so you'll see for every group of 3 in the DNA or the RNA, it's going to code for a single amino acid. Okay, so for example, I could give you, just kind of as a practice, if we had, let's say, 300 amino acids, which would be, definitely a typical size to a protein, even maybe on the small side. So if we had 300 amino acids and I asked you how many nucleotides, okay, code for that, you would just have to multiply by 3. And so then you would know that there are 900 nucleotides that are required, at least at the minimum, to code for that 300. Okay. So that's just a practice. Okay. So again, we've known about this genetic code really since the 1960s. If I go ahead and show you a picture of what that genetic code looks like, you can see there's a lot of redundancies in the code, meaning, for example, if you take a look right here in the genetic code, you'll see if one individual has CCU for their codon, somebody else could have CCC, I could have CCA, your neighbor could have CCG, but it's still going to code for the same amino acid, which is proline or pro. Okay, so that's why we say the genetic code is redundant, meaning we can, even in some cases we could have even a mutation perhaps that's really silent, that has no effect, because maybe the mutation takes place, and so instead of CCU it's CCC, and it still codes for the same amino acid, so it's not going to have any effect at all on that individual. You can also see here, it took a little while to figure this part out, but we also have 3 that don't actually code for an amino acid. They're going to be basically stop signs at the end of the gene that signifies that the protein has been completed. So we'll see these 3 stop codons towards the end of the lecture. And then we also have one AUG that's at the start really of the gene. So we always have this at the very start of it, and this at the end of it, kind of as our signal. So AUG codes for start, but it also does actually code for an amino acid. So you'll see methionine not just at the start, but sometimes you'll see proteins that have methionine as their amino acid at various places. Okay. The genetic code is also universal, which we've seen in our transformation lab. So we took this jellyfish gene here and we inserted it into a bacteria. And so since DNA is DNA and we operate off of a similar universal genetic code, our bacteria could read that jellyfish gene in its organism and follow the directions and produce that glow-in-the-dark protein that we saw. Just like here, they inserted it into the pig when it was an embryo, and so specifically you could see if we shut out the lights, we'd see the hooves and the snout glowing. They've done something similar with a very similar gene, the firefly gene, and used it to trace the development of a plant, a tobacco plant. And so we've talked a lot about genetic engineering, and we use the term transgenic specifically when we have moved a gene across species. So every time you see that trans, think that we're moving across to a new organism. Okay. So now we're going to get rolling on the process of transcription. Okay, so remember again, transcription is anytime you are going from DNA to RNA. Okay? So luckily, unlike replication, there's only one enzyme to know. Again, ASE tells you this is the enzyme here. Okay, so RNA polymerase really has two functions. Okay, so it is going to unzips DNA during transcription, and it's going to build RNA. That's why its name is RNA polymerase. And it's going to do some proofreading. It's not as great of a proofreader as we saw with DNA polymerase. But again, because the genetic code has some redundancies, it doesn't have to be perfect to get the correct amino acid put in place. Okay. So if we take a look here, first, before we talk about the promoter and these transcription factors, there are going to be some new base pair rules. So specifically, remember in RNA language, we still have adenine. Okay. We still have guanine. We still have cytosine. But now the difference is going to be, instead of having thymine that we saw in DNA, we're going to have the U, which is uracil. So where you normally wanted to put a thymine, you will put a uracil. Okay. So if we take a look at these three here, this promoter, transcription factors, and terminator, okay, so if we kind of label on this picture here, imagine here's the actual gene. Okay, here's the segment of DNA that codes for some kind of a protein. Okay, upstream of that gene, we're going to have a promoter region. A lot of times they call that promoter the Tata box because you will see a bunch of adenines and thymines in there. Okay, so upstream is the promoter, and then downstream of the gene, we can't see it here, but imagine downstream this way is going to be something called the terminator. Okay, so again, it's kind of signifying, going to give RNA polymerase a little bit of help in finding where the instructions are to make a specific protein. Okay, so RNA polymerase needs a lot of help, and so one of the things that's going to help RNA polymerase are these transcription factors. Again, these are also proteins, protein, protein, protein. And these transcription factors their job is to basically look for that Tata box and bind to it. Okay, so here you see 3 transcription factors, but then there are a few more show up here as well. And so this cluster of proteins gathers around that Tata box. And then again, that's the purpose of that is to help RNA polymerase, our enzyme, find where to begin. Okay, so once it binds, this is RNA polymerase here. And once RNA polymerase binds, you can see it starting to unzip the 2 strands of DNA. Okay, it is going to work in a 5' to 3' direction, just like DNA polymerase did. Okay, now notice this is only building on one side, okay, because RNA is a single strand, so it doesn't have to copy the DNA on both sides. Okay, so this is called initiation. Okay. Elongation, again, this kind of tells you the speed in which RNA polymerase can work, so about 40 nucleotides per second. So if you take a look here, again, it has unzipped the DNA and it started to build. So if it sees a T, it can still put an A, so we still find adenine in there. But if it sees A on the DNA, it's not going to put a T, okay? Instead, that's where it would use the U, okay? G goes with C. Again, that looks like another G, so another C. If it sees T, it's going to put an A. We have another T, A. Again, here's A, so this is where it would use U, and so on. Okay, so it's just going to basically be doing this. Again, 40 nucleotides per second, building and doing a little bit of proofreading as it's going. Okay. So it's going to keep doing that, and you'll notice right here where this single-stranded RNA is coming off,